German, I'm not sure. Some brilliant student, grad student, actually started working on the electron microscope. And so by 1939 or something, it was like, I think it was his dissertation maybe, but they were building an electron microscope, and by 1939 they did find the first virus using that using that technique. So it took about, maybe another 50 years between postulating that there is something out there that's actually even able to infect other, for example, bacteria or other cells, and that's very, very small, smaller than other microbes like bacteria. So it took about 50 more years, but by 1939 they identified the tobacco mosaic virus, and I think we have a picture in there, so I'll show you in a little bit. Just so you have an idea of how recent really viruses were discovered and the progress that's been made since. Here are a few things. So always remember that you come across that question. I mean, viruses, because the theory is they've been around for forever, absolutely forever. They can really infect any cell. So it doesn't matter if it's a bacteria cell, if it's a prokaryotic cell or a eukaryotic cell. They're not cells themselves, but they can infect anything. Plants, animals, if you go from the top, or from the more developed or higher organized organisms to algae, fungi, protozoans, bacteria, everything. So you can find them in any living cell or in any living organisms, because that's part of the definition of that. And just trying to figure out what's important on this again. You all know it's not considered completely like a living, or it doesn't have all the characteristics of life. Most of the characteristics, but not all of them, because it doesn't have the cellular structure. So that's one of the main distinctions between viruses and bacteria, and then eukaryotic cells. It's always active in the host cell, so that's where they replicate, that's where they develop, that's where they live. They're not, and I looked up a number for that too. We'll come to that later. But they're only able to survive a very short amount of time outside of a host. And survival is one thing, but when you think of life, if you can't reproduce in an environment, then that's really the main constraint. So they're unable to do that outside of a host. They usually have either. So those are really important ones. Another one is DNA and RNA. One of the two, but not both. So in our cells we have DNA, but then we have that contains our genetic material. The genes, the chromosomes have contain those, but we also have RNAs that. Remember, they help with the protein biosynthesis, and they're part of the ribosomes at the endoplasmatic reticulum that help produce that. So we have RNAs that transport information. So they only have either one, not both. And then another thing you should remember is that very basic, really, and we get to the structure in a minute, but they're not really metabolizing anything on their own. They're basically taking over a host cell and have the host cell do all the work for them. Size-wise, it's a big distinction. So the biggest ones around here, you would comes kind of close to a general size of bacteria, but a lot of them are a lot smaller. So that's why it took the electron microscopy to actually discover them. If you want to think of the structure of them, just imagine going again from the outside to the inside. You always find a cover and a core. So the cover protects the core, and the core really is mainly a nucleic acid, some matrix protein, and maybe some enzymes. But really, mainly what you're mainly concerned with, or what the virus is mainly concerned is protecting the nucleic acid molecule, which is DNA or RNA, and then you have a capsid and sometimes an envelope, and that's pretty interesting. That's a major distinction. You have some with a capsid, which is a protein structure to protect, but then you also have some with an envelope in addition to the capsid, and that changes how they can enter the host. But we'll get to how that happens and what the differences are between the naked and the envelope viruses in a minute. So that gets me to this slide where you see the major difference. So you have the capsid and the nucleic acid, and then in the envelope viruses you have the same structure, but you also have an envelope. And on those envelopes, a lot of times you find protein spikes that also help be recognized by a host cell and invade a host cell, and we get to that too. So nucleocapsid really is what you call it, because you have a nucleic acid covered or enclosed by a capsid that's made out of protein. And then you remember the two main structures, helical or cylindrical, so you form these cylinders, and that first virus that was discovered through electron microscopy was a helical one or a cylindrical one, the tobacco mosaic virus, so it was a plant virus they found. So you have the cylindrical nucleocapsid, and then you have the icosahedral or icosahedron. You have 20 sides, 12 corners, and what they call here fibers, but it is similar or identical to the spikes. I can't really find a difference between those two, and they help attach to the host cell, which is very specific, and we'll get to that too, how specific those structures are when it comes to attaching to a host. So talking a little bit more about we talk a little bit more about the viral envelope, and it happens mostly in animal viruses, and it's not something the virus produces per se itself. It's not producing that envelope. It's basically making the host cell to include this envelope into their own membrane, and so when it leaves the host cell, the host cell has already produced a lot of those proteins that are specific for the virus. The host cell produced something for the virus, and the virus takes it out on the way out, and has these spikes, and then with those spikes, it's able to attach to a new host cell and spread again. Maybe one thing that's interesting, or you should know, is if you think of the difference between a cell, eukaryotic cell, how much bigger they are than the prokaryotic cell. So I think in general, just to have an idea, I think a virus that invades an animal or a eukaryotic cell, like one of those more developed, bigger ones, or more highly developed, you would imagine maybe 1,000, 2,000 viruses leaving, so that when the first virus comes in and then replicates and reproduces, by the end of the cycle, when the virus leaves, there's probably 1,000, 2,000 virus particles assembled back and ready to infect the next cell. And for bacterial cells, because prokaryotic cells, because they're much smaller, you might see more like 200. So it's a tenfold decrease, but it makes sense because the cell size is different too. So viruses that actually use prokaryotic cells as hosts don't reproduce as much, but because of the size, the density may be similar. I don't know what the exact numbers are, but just so you have an idea of
You mean like per cell?
Yes.
Wow.
So because they're so little or so small and they're so basic, it's actually possible to, it's even possible for those cells to sometimes survive. But you can imagine if you have one cell and then you have, it's hard to say, what initially happens when you get an infection, say influenza, but you would imagine if every cell replicates or produces virus, and you get about 1,000 or 2,000 virus particles of viruses, and then they can again, so they would, out of one you get 1,000, 2,000 new ones, and they're able to infect 2,000 new cells, and then you multiply that by 2,000. It's a pretty fast spread, and that's usually what happens in an infection until it's controlled by medication, and we get to that in a little bit too, or by helping your immune system. So it takes a while for the immune system to recognize structures and to react to it, and so the earlier that happens, the less you're going to feel that effect. But the changes, and we'll talk about those too, that virus brings to your cells is really what creates the damage in bodies and cells and in tissues, and that could be more long-term or short-term depending on what virus it is. So this is that is the famous virus that they first, tobacco mosaic virus that they first discovered, and it's a typical cylindrical one. So the outside, the capsid or the envelope is obviously, we talked about it to protect the genetic material of the virus, but it also helps the virus to bind to cell surfaces, penetrate the cell, and supply the host cell with RNA, DNA to produce and some other genetic information to produce virus particles. And then we talked about those a little bit. We're going to go more into detail how these work. So those are the ones that actually parasitize bacteria, the bacteriophages, and they really do look a little bit like moon landing devices. So I always think of those as, for some reason, I always think of those as the typical virus, but that's really, those are the viruses called bacteriophages that actually invade bacteria. And a lot of times, which we're going to get to too, but just so you've heard it before, that actually can change the bacterial toxicity. So the virus in combination with the bacteria are actually more toxic, and that makes it really important for us to study viruses and bacteria and the interactions, because sometimes you have bacteria that are not as toxic, but in combination, when they've been invaded by viruses, their toxicity increases by a lot. So that's when it becomes important to the medical field.
So like the virus would infect the bacteria and they both go infect the host, or would this take place already in the host?
Depending on where they live, and I'll show you in a little bit how that could work, because not always you not always have an immediate reaction, so you wouldn't necessarily, because you could say, okay, if the virus invades a bacterial cell, it multiplies, and then you have a huge amount of viruses, and they destroy the cell. How is the bacterial cell even going to have any kind of toxicity? But sometimes there's a cycle where bacteria actually just enclose, and it happens in our bodies too, but they basically just keep the DNA or RNA of the virus and include it in their own gene or genetic material, and so through certain signals it creates more virus, or there are other instances where it just stays in there. It's like a latent infection, and so the bacteria might be infectious, or the bacteria that entered your body might already or you might react to it a certain way, and if it has the viral genome enclosed in it, it might just produce a stronger effect because the toxins of the virus are also expressed. So it's a combination of two genomes.
But that'd be different than like an opportunistic bacteria coming in after you already have a viral infection, right?
Yeah. So those are combinations of the two. And in a picture you'll see how, you know, you have basically the genetic material of a circular chromosome of a bacterial cell, and then an enclosure of a virus, or a shorter DNA or RNA piece in that. So it's different. Yeah, it's a combination of the two. Those are just examples. You can look at them in your book. What's so enclosed and non-enveloped. We did this one. He said that's important. It's either or, but not both of them. So you wouldn't find it in viruses. You wouldn't find both. And the number of genes really varies, but that's a given. You know, the number of genes vary for any kind of species or organism. You never. I mean, in a species, it's probably very very similar, but between species you'll always find variations. If you can have a tomato with a huge amount of genes or genetic material, and you have other organisms that seem more complex with less. So it all depends on how much really codes for proteins, etc. But that's going more into the genetic makeup and how it works. So really, what's important here, and you'll understand more of that if you don't already about the DNA and the RNA viruses. It's just either they have DNA or RNA, not both of them, and then also there's various ways, single-stranded or double-stranded, and then sometimes it's already there, so the RNA is already ready to read, so it can be enclosed in the host's genome and be ready to be read into whatever the specific virus wants to, you know, basically replicate or produce protein and nucleic acids for the virus, or it has to be converted if it's a negative sense RNA. But what's important really is that at the minimum they have genes to synthesize the capsid, the genetic material, and then usually when it enters, it's able to regulate the actions of the host, so it takes over. It doesn't just enclose or enter its genetic information. It really takes over the whole process in the cell, and then it leaves after the cell produced these components, and mature viruses leave the cell. And I'll show you in a little bit there's only two ways how the virus may leave the cell.
So does it normally end up killing the cell then?
In general, it would, or it does, but there's a lot of exceptions. The latent one is one, or we'll look at it. Sometimes the cells somehow survive. They can deal with basically having a phase of producing for the virus, and then the virus leaves. And if in cases they may be non-functional, they may look different afterwards, but they may be able to survive. So that's a possibility. In general, the cell gets killed, especially in prokaryotes. So they usually produce so much. If you have a smaller cell, so a bacteria infected with a virus, it basically produces virus until it explodes. Don't worry about this. Don't write anything down. I don't know why I have that here right now, but so they would actually lyse, and that means the bacterial cell is dead. This is just to answer a couple of questions. We talked about this one. So I was thinking Ernst Ruska and Max Knoll. Sounds German, right? I didn't look it up. Could be Austrian, but we never give anything to Austrians. It's like you and Canadians. we only accept them. They're like they're good at skiing and that's it. Isn't that Canadians? What else? Oh, hockey. Yeah. But then that changed too, because the U.S. bought all the teams. This is an old question. I was thinking about like the few things that we really that you ask and that I mean a lot of things that you ask and I don't know, but so in general we talked about at some point when we went through the eukaryotic cell, I think you had a question about how long it takes, and when you started looking into protein biosynthesis, we were wondering how long it takes from the nucleus out to producing a protein, and I think it takes about in like E. coli, for example, a pretty fast rate. So they can assemble 40 amino acids per second. And if you think of a protein like a typical one, say 200 to 300, which could be way larger or smaller, but then you'd have 5-6 seconds to make a protein. That doesn't include the whole thing. And the problem is when you start looking around for protein biosynthesis, you end up on bodybuilding pages all the time. Like, dude, how much can I? Yeah, they're all talking about like how much protein you have to take in to synthesize more muscle or whatever, and you cannot get to the scientific questions. You just end up in
bodybuilding land.
Yeah. So this is 1939. That's the next question we had, and then this one was yours, Dan, right? I was just looking it up, and I think that's important information. So, viruses cannot live or survive out of the host cell. They can live for a while, but they can't really reproduce, etc. So they're not considered out of the host or free living organisms, but they can survive for a while. And so, depending on the surface, I think really, stainless steel, it might be a little bit longer. But in general, you have a few hours. I think if you have a large amount from a donor, they think maybe 2 to 8 hours on stainless steel surfaces, maybe only a few minutes on a paper tissue. But you do have that time where, we talked about maybe somebody sneezing and you have water particles and that actually help, but the virus, without any kind of transmission or transmitter or vehicle, would still be able to survive for a little bit.
Why do you think it's longer on the hard surfaces? They need to just the other way around? Yeah.
Well, I don't know. I would imagine that.
For example, a grocery cart, it can live 24 to 48 hours.
A what?
On a grocery cart.
Well, that could be. This is viruses. I don't know if they're. You're thinking about this part here, influenza up here.
Yeah.
I would just imagine that, but I don't know how, what conditions this was tested in, but I would imagine just in softer or if stainless steel strikes me as, when it's clean, whereas soft tissue, paper, there's always more activity of other
I would think that'd be advantageous for the virus, though.
Too?
I think it would be helpful for the virus to have other stuff rather than just by itself.
Yeah, it would be.
Well, how come it survives less time?
12 hours on paper, 24 to 48 on.
That's what I mean. Like I think, I think there's just more, and I don't know, but I can imagine that's how I would imagine it. More other microorganisms, other enzymatic or enzymes from other microorganisms that actually help degrade the virus rather than on a stainless steel one. But like I said, I don't know what the conditions were for those. It's probably if they, for example, had a stainless steel that was sterilized forever, and then you take paper, you have a lot more, just because of the texture, a lot more possibility of other microorganisms sticking to it, etc.
But, and that's just for the influenza, right? Like other
Yeah.
viruses probably vary quite a bit.
Yeah, it varies quite a bit, but in general, I mean, this seems really long to
It
me. I
does.
would say, minutes is usually what you learn. And it really depends on, I mean, they say here, large amounts, depending on, I don't know what the concentration was that they started out with. So you wouldn't imagine on a, even on a grocery cart, I think bacteria are a lot more a problem, but you wouldn't imagine, even if somebody sneezes on it, the concentration usually makes the difference. So that's really what, if you have a higher concentration, maybe you have more, it takes longer for some of the particles at least to die. So this is, it took me 25 minutes to just go over what we already did. This is new. So if you've slept so far, you have to wake up now.
Dr. Pat, is there an air conditioning adjustment?
You could always take a, I've heard that this place is not good.
Oh, here it is.
temperature. I know, but I think it's automatically, it was really cold last time in here and warm in here. Did you have a bad night's sleep again? I think we can try, but they say it's automatically set. Does it mean 68 right now?
It says it's 68, it's not 86.
Yeah, I don't think that it's 68.
Yeah, it depends on the settings are.
I'll bring a thermometer and you guys can all take a guess.
Extra credit.
Nothing goes without credit. I read about this in like the welcome to the Vallejo campus, and it said, this is all said, and what you can do is you can turn this on if you want. Your whole table is gonna get paper flown around. But they said in the winter time they had little space heaters for every student because it was so cold. So don't take this past winter. That's what I heard. This could be good. You'd be the most alert student in the whole classroom. Better than missing it.
Right there. Wow.
Okay.
And then I go on my phone, right, and then I go,
You have till 25. Everybody can you still hear me?
Yeah.
So, when you look at the virus, we looked at all the different structures, and what you have to remember is really the most important part is these enzymes. So that you have polymerases, replicases, reverse transcriptases. So those are all to synthesize the material. Those are all enzymes to synthesize DNR and or RNA. The cell is going to do that. To copy RNA or to reverse produce the genetic material, and that means you have a DNA and you make RNA, so you have a reverse transcriptase. Just remember, these are all enzymes to help or to initiate and to make sure that the cell can produce the virus's genetic material, but there's no genes for synthesizing metabolic enzymes. So a lot of the materials, other enzymes that are needed, which aren't very many, is usually taken over from the host. So it takes over the host cell metabolic resources. say for the protein, I mean the proteins are usually in there, the capsid proteins, but there are proteins that it borrows from the host. And really quick, the classification, I don't want to go too much into that because it's not that important to me. It's more important that we at some point learn different viruses and different bacteria. But it really starts at the family level, so you don't go all the way up to, or further up the classification or the taxonomy. You basically have 3 orders, 63 families, and then 263 genera or genuses of viruses. And then the family name usually ends in viridae, so you have herpes viridae is the family of the herpes virus, and then the genus name ends in virus, so simplex virus or herpes simplex virus. And sometimes you abbreviate it like this. If you already come across them, you just see HSV-1, and you would know it's the herpes simplex virus 1. And for classification, I mean, I'll go over it one more time maybe when we if we talk about the first test. I think it's just important that you know how, what the different, how it's put together, how the species name is put together. And we can talk about this more. I didn't really, I gave you like full credit for half the answer because I think it was confusing how it was put in the test question. The difference between species and specific name and that kind of thing. But for viruses, just remember you mainly have families and then the genus, and that's how you identify the family, and that's the genus. And this is just I'm not going to get into that too much, so if you look at the classification of them, and if you say, okay, there's important, and that's why I didn't, this is just a table you won't see much, but it's mainly DNA viruses versus RNA viruses, and then enveloped viruses versus not enveloped viruses or naked viruses. That's the main distinction. And then a lot of times, if you look at the common name, it really has to do with what the virus actually does. So that's part of what the virus is named after. So if you have hepatitis, or what's a good example? Yeah, you just have it's a herpes virus. So you usually look at the name of the disease, or you look at what the virus does, and that goes into the name of it. Sometimes I think there's some, like the HIV, human immunodeficiency, so it has to do with the virus actually being involved in your immune system and what it does to your different immune cells. And I think we're going to talk about that one more. But I think it's more important to know a few rather than to know the classification, because when are you ever going to use it? It's more important, I think, to know what they look like, how they work, and what the main differences are in structure than knowing how they're named. So that gets me to the viral multiplication, and this is the most important differentiation I make here is. This is for, say, an animal virus. So this is either the enveloped or the naked virus that we're talking about, and then the bacteriophages, because they look like moon landing devices. They also have a way of entering the host cell that makes sense when you look at their structure. So we get to those in a little bit. So remember, this is for regular, this is the basic, this is the basic viral multiplication method or process. And this, for example, shows an animal virus. And so the first thing the virus has to do is absorb. So say you have a virus that came into your body. You would imagine, so you would ask yourself, number one is, why would it attach to some cells, or why does it affect some cells and why doesn't it affect others? Why would you have some that affect your lungs? Why would you have others that affect other parts of your body? It could be your liver, it could be anything. And so a lot of times the virus, or always what's happening is the virus has these spikes, has these proteins that can bind to specific cells. So they only bind to specific cells. The virus that enters your body may not find the host cell because it came from a cow or it came from a bird. So the virus always looks for, or can only bind to a specific host cell according to their proteins that you find on the outside. And that's true for envelopes and the capsid. So you would find that for both. Remember, they both have structures, proteins. And then it penetrates and tries to get the genome into the cell. And there's a little the next picture is going to show you how that works, and it makes it a little bit easier to understand. But it's really simple. It attaches to the cell. It somehow enters the cell and gets mainly wants to get its DNA or RNA into the cell. It needs to be uncoated to do that. So it means the nucleic acid that's protected by the protein shell or by the envelope has to be freed from the capsid. And then synthesis of all the viral components can start by the host cell, and then the viral particles will be assembled. So you have the host cell producing all these capsids, all these nucleic acids, all these other structures that the virus may need, and they are floating in the cytoplasm, and they get together through certain processes we're not going to get into because that gets really difficult, I think. And then they have to also get out. They have to release, or the virus has to be released. So it's somehow, just imagine a virus floating around in a body or in some kind of animal, in some kind of plant, and it needs to replicate. What does it need to do? It needs to absorb or adsorb. It needs to attach somewhere. It needs to penetrate, get the DNA in. It has to be uncoated first, and then it synthesizes, and all the synthesized material has to be assembled and then get out. So it's not, there's really nothing surprising in that process. And this is what it looks like. So in this case, you would have an envelope virus cell, or virus, not the virus cell, we'll see that, a virus, and the receptors here of the cell, so you have a host cell, and this is the cytoplasm, that's probably the nucleus, and so it attaches here, and then it can either fuse with the cell membrane, because if you think of it, we said that the virus picks up cell membrane on the way out, so it can actually fuse with the cell, or it can be put into a little vesicle, and then it moves into the cell. It uncodes, so the capsid is maybe digested or otherwise destroyed. It frees the viral RNA. Here's the RNA, and then this one is synthesized, replicated, and whatever proteins for the capsid, but also for other for example, the RNA synthesis or the DNA synthesis. All the other enzymes are produced, and here you have these different. Here you have the new RNA, because this is an RNA. So you have new RNA produced by the cell, by the host cell. You have capsimers produced by the cell. You have new spikes. And the difference here, and it just seems interesting to me that they actually are able to produce these proteins that are actually viral proteins, and they align them on the cell, or they are aligned on the cell membrane, and then when the cell actually leaves, it takes it takes some of this membrane with it. So when the virus leaves, it takes some of the membrane with it, and with it all the viral proteins that it needs.
What about the other stuff in the membrane? It doesn't get that?
It does, but it's mainly important. You mean like other membrane particles of the? Yeah, it does. Of the cell, of the host cell.
Yeah.
It does, but what it really needs is mainly the proteins that allows it to connect to a or to
Does it just
attach.
die off or go away, or?
No, I think it does have host cell membrane in it. It's not just cell membrane from.
So wouldn't that change it then, for when it's looking for a specific cell to go into that?
Well, it depends. I would imagine that, or I would say, if you have a host cell, so it first of all leaves the cell. If it does this successfully, it's in a cell that is a host cell. So it would go to the same kind of cell. So if this is a liver cell, it goes to the same kind of liver cell. And so it would, that would be number one. And number two, you would have the most, and there's a better picture, you would have the most dominant or prominent protein structures that the virus needs to invade the next cell, ready to attach to receptors. So what else is in that membrane is not as important to do what the cell needs to do. Natalie, we have a short quiz. Do you want to do this real quick, or you can wait until we have a break and then do it then?
Can I do that?
Yeah. So let's go through the steps real quick, and here's what I was talking about. Or if you look at those spikes, look how dominant they are. So they allow, in this case, it's a capsid that from a naked virus. It allows it to attach these specific receptors. Same thing here. So you have specific proteins on the virus that allow it to go there, or to actually enter that specific host. And initially it's a coincidence where the virus ends up, but it only can only enter if it's the right host cell. So if it's a hepatitis virus or a virus causing hepatitis B, it is the human liver cell. Or in the polio, it's the intestinal or the nerve cells. So it really depends, and some are more specific than others. So you wouldn't. A lot of times, what the medical field is worried about is, for example, that there's viruses that were strictly affecting, say, cows, and then all of a sudden some kind of mutant or some kind of slightly different virus form is able to infect us. And so how do we protect ourselves when we eat beef? And so there's all kinds of variations possible, but usually a virus doesn't affect your whole body always. It affects or it replicates, it multiplies in a specific cell. So it's like the virus's host range. And it all depends on what kind of receptors that specific cell has. So all your cells and all your tissues differ in what they accept, or what they let in and what they won't let in. And so one of the things I talked about is, if you go back to here, this can actually really happen. This penetration can happen in two ways. You can imagine that fusion happens because you have this bilayer that the envelope virus has, and so it's pretty easy with that bilayer to actually fuse with the membrane. And I don't know if there's a picture. It would look something like this. So you have an envelope virus, and it fuses with that membrane, and then releases the capsid.
at the surface, right? At the outermost layer, it kind of.
Yeah, so you would have the host cell here, and this is the enveloped virus, and you can imagine that these layers are. It's because the bilayer is not a structure that there is always movement in it anyway, and so kind of fuses together. Yes.
I want to ask about the spelling of. I thought it was absorption, but it's spelled A-D.
Adsorption.
Adsorb?
Yeah. So it's well, you could say it's basically attachment, absorption, but absorption would be.
It's with a D though?
Yeah, because the absorption would maybe be the what the cell does to it. You know what I mean? Absorption. It's attached. So let's call it attachment for now. Attachment to the host cell.
Okay.
I think what it does, when you get to the layer, it adds to it instead of actually ingesting it.
Okay. The next step is the engulfment.
Yeah, so in this case, if you do have, so fusion makes more sense for an enveloped virus because you can imagine that the membrane fuses. The membranes fuse of the virus particle and the membrane of the host cell. But then you also can imagine that we've talked about this before, and we're going to go into microbial nutrition, and there's endocytosis. There are other mechanisms, but basically an entire virus is taken in and enclosed in a vacuole or in a vesicle. So you would, because it binds to the receptor, the host cell is not rejecting it. The host cell is doing what it would do with, say, a larger nutrient molecule. It takes it in, and then there's two different ways. It either breaks the envelope, and then breaks the capsid, and it releases the nucleic acid. Or it actually, if you have a naked one, for example, it attaches to the receptors, it builds this vesicle, it comes in, and really that vesicle stays intact, and only the nucleic acid comes out. And then the process starts for replicating that virus.
Wait, so if it goes in naked, it gets the capsid, and if it goes in with the capsid, it loses?
No, it's just, these ones would be identical. It's just different ways of,
Okay.
this is not a capsid, this is just basically a vesicle, a transport vesicle that brings it in, but it doesn't necessarily, it's released from the vesicle, the RNA is released, it's not actually degrading this part. It probably eventually is, but the RNA is already out for replication. So the most important differentiation is fusion, where the membranes of virus and host cell fuse, and then for the naked ones, for the naked viruses, you usually have a vesicle, and it's either digesting the capsid and releasing the RNA, or it's just releasing the RNA. And remember, that's all for viruses in cells, but prokaryotic cells. So for bacteriophages, it looks a little bit different. Not that different, but there's a slight difference.
So can you clarify the difference between those three?
So this one would be fusion, and you have, like in this case, you have an enveloped virus, and so the envelope with the membrane makes it possible for, we talked about the bilayer, the membrane bilayer that you have in regular eukaryotic cells, and so when that happens, those membranes fuse together, and that's how the virus is able to enter the cell. And then if you have naked ones, like this one, for example, or this one, you usually find endocytosis. So they don't fuse with the cell absorbs them or engulfs them, and then either digests the capsid and releases the nucleic acid, in this case DNA, or it just there's no that stays intact, at least for now, and it's just the RNA DNA is able to leave out of this vesicle on its own. So see here, you still have the virus intact, and all it does is basically infect the cell with its RNA. So now it's in the cell, the RNA goes out, and the host cell starts producing virus. So it always ends up being the same thing. You always end up with the free RNA, free DNA. You always end up with the nucleic acid in the host, which is what the virus is that's what the virus is mainly concerned about.
What was the bottom one?
These are just different because of how the vesicle is, how the vesicle that has the virus in it treats the virus. So either it's digested and it's free, or it stays in this vesicle for a while and just the RNA is free. So it's just a difference of, is that capsid?
The bottom one is a naked one also, right?
Yeah. So one other distinction is, so the replication of that genetic material happens in different places. If it's a DNA virus, it usually happens in the nucleus, and it makes sense. If you want to think about it that way, you can say, well, that's where DNA replication synthesis happens in a regular cell, so that also happens for the virus. And then for RNA, it usually happens in the cytoplasm. So that's where and that would be also the case, I mean, in that you have RNA in those cells assembled and replicated in the cytoplasm and not in the nucleus. But that's just a main, or the difference between if you look at a DNA or RNA virus. And then we talked about this a little bit, but I don't want to confuse it too much with that. Is just that that positive sense just means it's already ready to be used, whereas this negative sense RNA must be first converted. And it's just because some viruses decided to go the complicated way and produce negative sense RNA, we have to learn about it, and it has to first be converted. And so you need different enzymes for that. But really, I mean, you need to know some of this, obviously, but it's mainly of importance that you understand that how the virus takes over control and how it replicates its own genetic material and what happens with it. And then it has to get out. So once the 2,000 virus particles are ready for assemblage, so you can imagine you have a cell, a eukaryotic cell, pretty big cell, and there's all these particles in there, and they have to somehow be assembled, and so that happens in the cell. And then there's just like the cell was taken in through endocytosis, you have exocytosis, meaning it's down here. It basically allows, and this is the reverse. This is basically the reverse process. In this case, you have the cytoplasm, and you have these RNA pieces, and you have capsids, and they assemble together and start going towards the cell membrane, and then they pick up some of this, what we talked about, some of that membrane of the host, and through exocytosis leave the cell, and then they look like this, and they have proteins that allow them to attach to the next host cell. And if you're already in the right environment, if you're in a specific tissue, you won't have to. The virus won't have to look very long to find a new host cell. The other way, and that's only, and keep that in mind, the lysis usually happens can happen in these cells, but it usually always happens in bacterial cells. So lysis would be for non-envelope viruses, and for more complex or for some complex viruses, the cell dies and it ruptures. And a lot of times it's just because of the accumulation of toxins and the accumulation of products from the virus make the cell die, or it really becomes there's too much virus in there, and it just ruptures and the virus is released that way. I stressed this a couple times. I left it in. Okay.
One,
Or just small.
Yeah.
Okay, balls.
how can they be?
Either one. Probably 29 and 5 minutes. Is that possible? After the viruses, we can take a longer break. I just want to get through those before we, or I want to cover them pretty much all together. So if you want to take a short break, want to do the viruses, maybe we'll be done with them by like say 10:30, and then we have a little break, and we have like an hour to get into the next chapter. I'm not going to have anything to ask you for the next chapter test. With the speed I'm going. Yeah?
Yeah.
Okay.
And you're gonna have the study guide on Wednesday?
Yeah. When?
On Wednesday?
No.
Like last night?
Tonight.
I mean, you guys did so well in the first one.
Really?
Really?
I think so.
I didn't.
You should